As we continue to talk about different types of traits, I want to introduce a new one today. Up to this point, we've talked about traits that operate under the idea of complete dominance, incomplete dominance, codominance, and multiple alleles. And we want to get to a fifth option today. What you're looking at here is a picture of somebody's chromosomes. That picture is called, as it is labeled down below, a karyotype. What you're seeing here are 23 pairs of homologous human chromosomes. And if we look at the next picture as well, you will see, in essence, the same thing, but with one important difference. There are not 23 pairs of homologous chromosomes in this particular person. Instead, there are only 22 pairs. There are 22 pairs of homologous chromosomes, but one pair, located right here, of non-homologous chromosomes. Now, there's nothing wrong with this person. They are simply male. The previous picture was a female. So females have 23 pairs of homologous chromosomes. Males only have 22, with then one non-homologous pair. And we can look at the difference between that 23rd pair of chromosomes here. Ignore the labels with the letter B for a second and just focus in on the fact that a female, pictured here on the left, has two of what are called X chromosomes. They are a homologous pair. If you remember that word, it means the same, but not identical. In other words, if there's a gene here that deals with a certain trait, there is a gene here that deals with that same trait. Now, they might not be identical. This person might, for instance, be heterozygous for that trait, but they're dealing with the same trait. When it comes to a male, pictured over here on the right side, this is a non-homologous pair of chromosomes. You can see just based on the size that they cannot be the same. So if there's a gene for a trait right here, there is no partner over here. This Y chromosome, and again, a male has an X and a Y, this Y chromosome carries completely different traits than are present on the X. In essence, this Y is what makes a male a male. Mostly the traits that happen to be on there deal with the production of testosterone, the hormone that makes a male a male. And there's not a whole lot else there. You can see it's a very small chromosome. We can go off onto a little bit of a tangent here for a second just to address something that you're probably a little bit familiar with already. You've probably heard the fact that in a male-female situation, it is the male, it is the man that determines the gender of a baby. And by looking at this picture, you should now see why that is the case. You know that meiosis occurs in order to create sex cells. And so if we look at the female, you know that this pair of chromosomes is going to be divided up during meiosis. One of them is going to get pulled in one direction. The other will get pulled in the other direction, and they will end up in different cells. But all that means in this case is that each egg, each female gamete, will have one X chromosome, but they will all have an X chromosome. That is different than in the male situation. When the male goes through meiosis, this X chromosome is going to get pulled in one direction. This Y chromosome is going to get pulled in the other direction. And so half of a male's sperm will be X sperm. They will have an X chromosome in them. The other half of his sperm will be Y sperm. And so it all depends on which type of sperm fertilizes an egg. If the sperm that is an X sperm fertilizes an X egg, that baby will have two X's and therefore be a daughter. If a Y sperm fertilizes an X egg, then that child will have an X chromosome and a Y chromosome and therefore be a son. So again, that's just a tangent off of what we really want to talk about here today. As I said, we'll come back to these labels that I have here. The picture that you see here on the left is an X chromosome and on the right a Y chromosome. It's just showing you some things that are found, some of the traits that happen to be found on these different chromosomes. you can see that the X chromosome is big. It carries a whole lot of traits, and that the Y chromosome is small and carries very few. And I told you mostly what's on the Y is the production of testosterone. I don't know why they chose these particular traits to feature on this picture of the X chromosome, but they did happen to choose traits that are all negatives or sort of bad in some way or another. We're going to focus in, as we deal with this idea of traits on the sex chromosomes, we're going to deal with this idea of red-green color blindness. That's probably something that you're all familiar with, and you probably even know somebody who is colorblind. So with that in mind, let's go back to this picture right here. As I said, you probably know somebody who is colorblind. So colorblindness is what's called a sex-linked trait. And if you want to be even more specific about it, it's an X-linked trait. As we talk about these types of traits, sex-linked, we are in essence really going to deal with the X chromosome because, as we said, the Y chromosome, although there and present and determining that a person is a male, doesn't contribute to a whole lot of different array of genes. And so we're going to deal specifically with the X. And our example on the X is one of colorblindness. So colorblindness is a recessive sex-linked trait. In other words, it's indicated by the small b in the picture. The large B that you see on the left here is the dominant form of the trait, and it happens to be for normal color vision. Now, when I said that you probably know somebody who's colorblind, I want to be more specific about that. You probably know somebody who's colorblind, and they are a male. And maybe by looking at what I have here, you can see why. Let's look at the female first. This female has one gene for normal vision and one gene for colorblindness. What will be the phenotype of that female? Well, she will have normal color vision. Why? Because she's heterozygous. She has a big B and a small b. And that big B, that dominant allele for normal color vision, will of course overpower or mask the recessive colorblind gene. She will not be colorblind. But let's look at the male here. The male, like our female example, only has one gene for colorblindness. But of course, he only has one gene at all for the vision, for color vision. There is no second copy of any gene. So by inheriting just one gene for colorblindness, the phenotype of this male is that he will in fact be colorblind. There is no second copy. And so that's what's unique about sex-linked traits. It's not so unique for the female portion of things. It's unique because of the male aspect here. There is only one gene. There's two in everything else that we have talked about up to this point. I've always said, hey, you inherit one gene from your mom and one gene from your dad. Well, when it comes to sex-linked traits in a male, that's not true. This male is colorblind, and he's colorblind because he inherited just that one gene for colorblindness. Just something to throw in here, again, a little bit of a tangent, but since we're talking about colorblindness, you couldn't necessarily look at somebody's gene. You can just do a simple test to find out if somebody is colorblind or not. Now, there are different types of colorblindness. Some of them are more common than others, but what you're seeing here is that the idea of colorblindness is that people, it's not that they see in black and white, it's not that they don't see color, they simply can't differentiate between certain colors. So depending on what type of colorblindness somebody has, they might not be able to see the number 7 or the number 13, 16, 8, 12, or 9, just couldn't separate those different colors. When it comes to this idea of sex-linked traits and the doing of Punnett squares, we are going to have the situation that you see here. So I have X because the trait happens to be on the X chromosome, and I have a superscript with a capital letter. I just happen to choose the letter B. to represent the dominant allele of normal color vision. We have an X with a superscript lowercase b to represent that recessive allele of colorblindness. And we do need to have the presence of the Y chromosome because we do have males and they have to have that Y chromosome. Notice that there is no superscript here. This trait of color vision or lack of color vision is on the X chromosome only, not the Y, even though the Y is present. So in terms of the different genotypes we could have, we now need to differentiate between female and male. Up to this point, I don't know if you've noticed at all, I've never talked about a mom and a dad. I've always said parent 1 and parent 2. That's because it didn't matter the gender of those parents. Of course, one of them was male and one of them was female, but it didn't matter who was who. Now that we're talking about sex-linked traits, it does matter. And so in terms of the female could be homozygous dominant, have normal vision. That female could be heterozygous and have normal vision. Or, and we didn't mention this, a female can be colorblind, but to be colorblind, she has to be homozygous recessive, has to inherit 2 copies of the gene for colorblindness. Just to stick with the female genotypes for a second, let's go back to the heterozygous situation here. There's a genetic term that you've probably heard that applies in this situation, and that term is carrier. Somebody who is a carrier is somebody who carries some sort of recessive, I'm going to say negative condition. So this woman here, although having normal vision herself, is a carrier of colorblindness. In other words, she's heterozygous. The male, on the other hand, there are only 2 possible genotypes. Either got X with the dominant allele and a Y, and therefore would have normal vision, or X with the recessive allele, and therefore would be colorblind. Here's an example of a Punnett square to the right. You can see we have a father here along the top with normal vision. We have the mother here to the side. She too has normal vision, but notice she's a carrier. She's heterozygous. And if we do the proper cross, we can really differentiate here left side versus right side, because to the left side we have children who are going to be daughters, and to the right side we're going to have children who are going to be sons. Notice both daughters, normal vision. Despite the fact that their genotypes are not the same, they both have a phenotype of normal vision. On the right side with the sons, we have a son here who would have normal vision, but we have a son here who would be colorblind. For a little practice, I'm going to give you a real scenario here. This happens to be my family. So I'm giving you the genotypes, or at least enough information for my 4 grandparents. And so we have at the top my maternal grandparents. That means my mom's side of the family. My maternal grandmother, uniquely, is colorblind. She is a female, but yet she is colorblind. So you should be able to figure out her genotype. My grandfather has normal vision, my maternal grandfather. And then we have the paternal side of the family. So these are my dad's parents. And I give you a genotype for my grandmother and enough information for my grandfather. So you have 3 Punnett squares set up here. What I would like you to do is to cross my maternal grandparents, and I want you to figure out the genotype of my mother. Use another square to cross my paternal grandparents and figure out the genotype of my father. And then I want you to use this 3rd Punnett square over here to cross my mother with my father, and I want you to see if you can figure out whether I have color blindness or not. Actually, I want you to figure out the percentage likelihood that I am colorblind, and then I want you to email me the response. Give me a percentage. What is the likelihood that I am colorblind?